So before PCR, the way you would do cloning was you had to rely on the existence of specific restriction sites within an existing DNA to clone it. There was no amplification. So like if you wanted to clone a gene, I guess the best way to explain how much PCR changes the way you do genetic engineering is this. Before there was PCR, the way you would clone a gene is you'd start with the organism that made whatever functionality you had, that you think has some gene that you could put into a bacteria or mammalian cell or whatever and would do something. And you would isolate the DNA and you'd chop it up and you'd put all those DNAs into a plasmid and then you'd screen those plasmids to find one that had the right functionality. So it was a very laborious process to clone a gene. The way you clone a gene today is someone sequenced the genome already. Pretty much any bacterium, that's not quite fair to say yet, but many bacterium have been sequenced right now. So there's a very good chance you'll already have the DNA sequence for the thing you want coming from some database. Maybe they sequenced the whole genome, maybe they just sequenced a few genes from that organism. But very often you can get the DNA sequence for the gene you want. And then you can design these oligos for the PCR. There are like 20 base pairs on each side for that sequence. And you never had to have the physical DNA to design that experiment at all. But then all you need is a source of the DNA and you can amplify it out. So that's the way cloning has been done since around 1990 or so. And that's not totally fair. Using PCR to clone genes has been done since 1990. The sequencing in addition to PCR, where you could go straight from a database to the PCR and having just all this data about what things are and being able to go directly from that sequencing data to the PCR reaction, that's more like 1998, 2000 on. But I mean, that's how it's always done now. All right, but the new sort of way of thinking is the DNA total synthesis, gene synthesis technology. And what you're doing there is no physical source for the DNA. You have just databases of sequences, and you design out the gene you want on the computer, and you put that sequence as text, H's, T's, and G's, and you send it to a company, and they make that DNA for you. So the real organism was never in the picture. You just had the sequence that was from that organism, and you make the DNA. And so that's kind of the future. And I would say the use of that sort of approach right now, we sometimes do it and sometimes don't. And it's really just a cost calculation, a cost and time calculation as to how hard it would be to acquire the source organism's DNA to clone the thing out versus how much is it going to cost to synthesize the gene. But there's a lot of effort to reduce the price of gene synthesis, or total synthesis, as I prefer to call it, so that price gets down to the point where, in the future, what inevitably will happen is that price will come down to a point where it's never cost effective to clone a gene from a source organism. And when that happens, you won't ever have real organism DNAs in the lab at all. You'll always just be putting in the computer and ordering it from a company. And what that company's doing is they're synthesizing that DNA chemically based on something called phosphoramidite chemistry. So there's no organism involved, and they build it up base by base by base, and they build short single-stranded DNAs. And then they use a bunch of in vitro methodology to extend those small DNAs into the larger piece of DNA. And then they send that back to you after inserting it into a plasmid. There's something useful in there.
Useful stuff in there.
I have to reorganize that thought.
So I guess that's a good spot to go into your definition of parts and composite parts.
Ah. Okay, so there are two distinct ideas when it comes to parts. So first of all, you have something that the term basic part is used to mean both a primitive part and a fundamental part. I'll distinguish it this way. So you have basic parts, and there are two other words that sometimes get used to describe basic parts: a fundamental part and a primitive part. And the distinction between these two things is that the primitive part means a DNA sequence that cannot be reduced into a simpler sequence in the context of a standard assembly process. So something like a BioBrick standard assembly. You can't break this little DNA up into something smaller that could be assembled by standard assembly. And so standard assembly-based basic parts like you find in the registry get assembled into what are called composite parts. So composite parts always refers to assembly. It has nothing to do with functionality. And the idea of the primitive part also has nothing to do with functionality. I mean, the thing might not encode anything. It just has to do with assembly. All right, so the other idea here is that of a fundamental part, which, you know, sometimes when people say basic parts, they mean more like fundamental part or more like the primitive part. They use it both ways. But the idea here is that you have DNA elements encoding the core functionality for doing genetic engineering. The elements such as promoters, ribosome binding sites, the coding sequences for proteins, terminators, origins of replication, things like that. It's all the basic elements that make up a genetic program. And you're piecing up devices from basic parts. So device refers to functionality, not physical assembly. And in fact, many people would view devices as something that can exist on separate DNAs. They don't even have to be on the same DNA. Composite part implicitly means basic parts strung together on a DNA. Device could be 5 different DNAs or, you know, distinct sites of the genome. It says nothing about where things are. It has to do with function. And the distinction between the basic part and the device is function. The basic part doesn't do anything. A basic part is too small to do anything. It only works in the context of other basic parts. So there's a composition issue where you have to put basic parts together into a composite part that then acts as a device, meaning it has some sort of higher order activity. So the simplest kind of device you could have would be an RNA generating device or a protein generating device. So all you need to make an RNA generating device in E. coli would be a promoter, an RNA encoding gene, and a terminator. So that's about as simple as it gets. To make protein, you just add a promoter, a ribosome binding site, a coding sequence, and a terminator. And that thing will make protein. So that's functionality. You're changing the composition of the biochemistry of the cell by making that protein. So it imparts a new phenotype. And devices can get really complicated. I mean, I don't know that we've seen yet really complicated systems in terms of synthetically derived things, but some of the things that were done last year in IGEM, like the toggle switch that won IGEM last year by the Peking team, that was a fairly complicated device. I mean, it has several levels of hierarchy in terms of behaviors going on within it. So these things can get pretty fancy. But, you know, if you look at natural systems, where in natural systems, you know, they're not engineered by anybody. They're just naturally derived things, and people tend to refer to them as, you know, loci in a genome or modules or cassettes. They don't use the word device. But you tend to get this effect in biological systems, particularly bacteria, where all the genes associated with some really complex biological activity are all clustered together. So if you look at, for example, bacteria that do photosynthesis, you'll find all the genes necessary to make all the pigments and the light harvesting complex, everything associated with being able to use light to make energy for the cell, biochemical energy. You'll find them all clustered together in this 40,000 base pair chunk of DNA. And in a sense, that's kind of like a device, a really fancy device. You have all these parts encoding lots of proteins that are all interacting together to make this higher order biological activity, right? But it's just not an engineered thing. But the existence of those things tells you how complex you could potentially make a device. But, you know, where we stand in terms of our ability to make complex devices is just nothing even remotely close